Okay, so here we are with Dr. Michael Kladde. Thank you so much for joining us today, Dr. Kladde. Dr. Kladde's scientific focus is the study of transcriptional regulation and chromatin structure / function. He has an accomplished record of developing innovative methods to answer formerly intractable questions. He devised the first strategy to detect long-range genomic interactions. It pioneered DNA methyltransferases as reagents for mapping nucleosomes and transcriptor factor binding, and his discoveries have led to the development of mammalian MAPIT, the first single-molecule integrative means of detection of CpG methylation, nucleosomes, and TF binding. Targeted MAPIT detects rare clinically relevant epigenetic states, such as in cancer stem cells, and has uncovered a novel mechanism of chemotherapeutic resistance to temozolomide in glioblastoma. Dr. Kladde's team recently identified a novel hub of transcriptional activators that regulate MLH1 and other MMR gene promoters, which has profound implications for the reversal of glioblastoma chemoresistance, as well as the genesis of neoantigens, a biomarker of favorable cancer immunotherapy outcomes. Dr. Kladde has a 23-year record of accomplishment, solving important biological problems as principal investigator, co-principal investigator, and co-investigator of many funded federal, state, and local awards. So thank you so much for joining us today, and I will go ahead and open it up to students' questions. And if folks are a little shy, we can start with one from the list, but let's see if anybody has a hand or a question to start us off. All right, and if we need to get warmed up, we can go to the list.
I can go. I'll pick one here. About
okay,
one
go ahead.
Fidel Trejo Ortiz. I thought that was a good question. How did let's see, do I have any advice on how I came to terms with what I wanted to learn and study? Was it a quick decision, or did it take a while to discover my interests? And what are some ways to discover those interests? So I think mine's a somewhat atypical sort of situation. So I think I got involved real early in research in school as an undergraduate at UC San Diego. And I was working on it was gene therapy early to try to figure out gene disorder in Tay-Sachs. Very different thing. Main thing is just get exposed to some areas and see if they take root. I thought that was what my career was going to be in. I'm one of these people that every area of biology sort of excites me. And it's sort of been one of my, I think I would call a weak point too. It's hard to stay focused for me. I like to do everything excites me and interests me. All right. So then I went on to graduate school. And there most graduate programs, there are a couple different ways you can enroll for PhD doctorate. One of those is you'll go in directly into a lab because you've already decided that's what you want to work on. You're mature, you figured it out, maybe as an undergrad, you got real excited by something you read, and you go straight into a lab. But that's sort of the non-typical track. Most graduate programs you'll enter and you'll do a series of what's called laboratory rotations, much like a doctor does in medical school. And you go to work in 3 or 4 different laboratories, and you get exposed. You're looking for not only the area, I think it's pretty hard. You can get from the literature that the lab's published, talking to people in the lab, you can get an idea what the general area is. I think there it's a little bit more, instead of content focused and what questions are you excited about, that's important, but you're looking for an environment that fits your personality. And they're looking at you to see if you fit their personality. I mean, some labs are very large, what we call sort of pejoratively a factory lab. You can get lost in the mix. If you're a student that likes a little bit more hands-on attention, I wouldn't recommend that kind of environment. And then the opposite is you go to an assistant professor's lab. They're just starting out. You're going to get a lot of hands-on. Now that's a negative for some people. Depends where you are in your learning curve and your maturity curve, right? So yeah, I had enough because I did 2 years of research after I was an undergraduate. at the VA working on insulin receptor action, published a couple papers. I did it because I really wanted to make sure it was a good fit for me. And I wasn't in, as what I see common today, a lot of students seem like everybody's in a rush. They're not willing to really see if it's a good fit. They want to get through it. Oh, I just discovered one thing in my PhD. I want to graduate now. No, I think you want to build up a body of good knowledge and become really well trained in an area and really figure out what you want to pursue. And there's places, you know, picking your PhD lab, the number one thing I'd say is learn how to ask good questions. Science starts with good questions. And said differently, you got to learn how to frame hypotheses. And I think that's the most important thing to get out of graduate training. You can switch complete gears and go to something entirely different after you have that PhD under your belt. And I did, well, I didn't really do that. So I started off and I fell in love in graduate school right away. Getting back to the question that was asked, I had no trouble. Well, I did 3 rotations and I went in this one lab. I liked the style. It was a larger lab. I wouldn't call it a factory lab. There was a lot of hands-on, but I really gravitated to this one project that involved chromatin. Everybody was working on what we call transient transfected DNA. You take DNA from a plasmid from E. coli that has no proteins associated with it. And then they transfect it into cells and it would go in, and who knew what was, I mean, those are useful experiments, but I think I recognized real early that that wouldn't give us the full picture of how genes are regulated in this field. We didn't even use the term epigenetic. It was there, epigenetics, but everybody does a catch-all now on gene regulation. It's epigenetic regulation, which is fair, but there are purists that apply the definition kind of in a only certain context. But I just fell in love with that. I did my PhD in it, picked my project right away after the rotation, settled in, and then I did a postdoc in the area, and I've been working in it ever since. So I think that's actually unusual, extremely unusual. Most students I see, postdocs that have come to my lab, they typically didn't work in that area. So it's not really, I wouldn't say, maybe if you pick it real early, it could be an advantage. But I've had other students, like I've had great PhD students show up in my lab that had a physics background. And people that come from a different area that have a different way of thinking, that's fantastic. Because, I mean, I could think that student got me to think about single molecule stuff, which was completely unheard of 20 years ago. And we started working on it. Matter of fact, you know, there were plenty of times I've taken student ideas and I just run with them. I took out a whole aim of a specific aims of an NIH grant. My first one, as a matter of fact, I got as an assistant professor based on conversations with him. And we worked that in, and that became his dissertation, and he got his doctorate in it. And I got my first NIH grant from it. So I think it was pretty exciting, I think, for me, that I've had a lot of input from students. I have an undergraduate that just joined the lab. He's very driven. He's done a lot of research in one lab, comes to me, takes a lot of initiative, likes to read, has a voracious appetite. And he just emails me out of the blue one day and just says, hey, I don't know if what I'm saying is going to work or not, but it was really great. And I looked at it, I thought about it more, and I am literally, when I get off this call, I'm going to go back to writing a grant. I'm submitting on that idea. He didn't have all the details there, but he also was, he's fairly fearless, and he wasn't scared to put himself out there. Even though I'm a person and an expert in the field for 35 years, I mean, and I don't know if it's because I give him that kind of security that he feels he can broach those topics with me, or I don't know what it is, but it's part him, maybe some me, mostly him. He's the one with the idea. It's great. So does somebody, let's see, we got some in the chat now, so we can go to something there.
Yeah, I can, if we want to go down the line. So the first one from Federica is asking if you've had a favorite research project over the years.
Favorite research project? Oh yeah. Oh yeah. Let's see. That's a good question. A favorite. A lot of exciting ones. Favorite that stands out. Yeah, I have one that I would say it's sort of the first time for me in my career. I had a favorite project that I would definitely call something that had me riveted. And it's become a first time in my experience, a tragedy now too. So, and this is a lesson in documentation, folks. Okay, so we started a project and it was another undergraduate and we were using CRISPR not to cut and gene edit and modify DNA sequences, but there's a whole other side branch that's come off of the CRISPR technologies that you can inactivate the nuclease activity, and you can use it to either modify epigenetics, or you can use it to change gene expression, or you can use it for diagnostics, really rapid in vitro diagnostics. Like we have a colleague, Dr. Jane in engineering that developed a rapid COVID test with this. Anyway, so it was a really great project. So what I do typically in our work is some of these tools that Dr. Watson mentioned is the way you figure out how a gene is regulated is by its chromatin structure and how it's packaged. The DNA is packaged with where nucleosomes are. And then another layer is where DNA methylation is. And then the third layer is how there's a lot of the amino acids in the histone tails that make up the nucleosomes. They're subject to a complete array of different chemical marks that push genes toward more active or less active. All right. And so going back to what I've sort of been real excited about, my favorite thing I think we've developed sort of in the past was we use the DNA methyltransferase to mark. If you have DNA and you imagine proteins there, imagine a canvas, and proteins are sort of like grease. So imagine you put grease here, that's a nucleosome, grease there, there's a transcription factor. And now you spray paint with something that you leave a mark that you can count. The classic way you do it is you use what's called like a DNA nuclease, like DNase 1, and it cuts DNA. And then we have all these next generation sequencing methods to count cuts. The problem with that whole strategy is it takes DNA and it literally cuts it in all these little segments. And if you think about different cells across a population, you can't tell if it came from cell A, B, C, D, up to thousands or millions of cells in your experiment. So you can't put it back together. So what we've done is I took that out, and probably my favorite thing I did was we used the DNA methyltransferase as a relatively innocuous way to mark sites in DNA that don't have proteins bound, or conversely, I could then deduce or infer where the proteins are bound. And we use the DNA methyltransferase that puts a little methyl group on, and then we have all these ways to detect it afterward. That's really exciting. I was really happy, great accomplishment. I'm very happy. It's sort of been my bread and butter and got me all the success I've had at Texas A&M and now here at UF for the last 15 years. But the problem is you take out cells from, let's say, a patient or something, you need quite a few to do the method. That's what we call a limitation of sensitivity. And then the next thing is then I paint it. Then I have to isolate the DNA. I have to go through preparing the DNA. I have to make it a library. Then we have to put it on the machine for next generation sequencing and get that data. And then there's a long process of bioinformatic analysis of that very large data set. And all those steps add up, and it takes a lot of time. So the project I'm probably most excited about, but I'm a little bit bummed to say the least, is we had used a CRISPR way, and there's an enzyme, a CRISPR enzyme called Cas12a. And one of the Nobel Prize laureates, Jennifer Doudna, that got this, I think, a year or two ago, they got the Nobel Prize for the CRISPR machinery. She found that what it does is when it binds DNA and it cuts and leaves a double-stranded cut, that activates the enzyme to do a very hyperactive, promiscuous, off-target cutting of single-stranded DNA. So you could just put in a fluorescent reporter, and if the enzyme binds to where it needs to, then it cleaves this molecule and it increases fluorescence very dramatically and very quickly. So now instead of requiring 2 weeks, if you had a well-oiled machine to do all the next-gen sequencing MAP-IT pipeline that we've developed, now I can detect open versus closed chromatin, whether something's bound by a protein or not, literally in 3 hours. All right, so now here comes the tragedy part of it. We had the enzyme prep. Somebody added something to it that isn't documented, and it was a magic ingredient. That's the only thing I can tell you. And somehow, even other aliquots from this lab that we've done, we've bought commercial sources of Cas12a, we've gotten other preps that were from the same exact lot. They were frozen down and prepared at the same time. And we compare them side by side. The original tube works. The second one that's supposedly the same doesn't. So the bottom line from this tragedy is document your stuff very carefully, because you might do something you don't think is important. And now we have a lot of troubleshooting we have to do to try to get back to where we were. I literally had the entire invention disclosure in UF's Office of Tech Licensing. I was ready to hit submit probably in a couple days, just letting it stew, proofread it, and we couldn't reproduce it. And so I never submitted that because it's not worth pursuing till I have it working again. And so that is my favorite project right now, but I'm a little stuck. So I've got other favorite projects that we'll pursue.
Wow. Jump into another question. So the next question is about, a challenge and a regret, and that kind of touches upon a major one, I think. So maybe we could go to the next question, which is about, Katie asks about work-life balance in a PhD and beyond. How do you maintain some sort of balance?
That's a great question. All right. Wow. Yeah, I think maybe I would regret, I think my work-life balance. If you asked me that in the beginning of my career when I was a PhD student, I had good roommates. I met roommates my first year in grad school. We decided to then become I met them the first year. I had a studio by myself the first year. I met other students that were of like mind, great roommates, become lifelong friends. We moved together. So I had a good social outlet, but man, I worked hard. I would typically put in 80 hours a week as a student, and I'm not exaggerating. I would often see the sun come up. There were reasons for that, though. We had to share pipette mint literally in my lab. We went through a funding little crisis for a while, and it was just easier to work. I'm a night owl anyway, and it was just easier for me to work across midnight into 4 a.m. So I wouldn't have conflict trying to get equipment access and stuff. But I think one regret is that I think I could have benefited a lot. I've become a real proponent lately as I've gotten promoted and up through the food chain, so to speak, and now full professor. I think I have a much better understanding of how important it is to have that. And I've even let myself recently, I think COVID was a real good example. We weren't allowed to go to gyms. Everything was shut down. I stopped going, and I just put that aside now for 2 or 3 years. When you get older, you learn things. Bodies at rest rust. And like I started having very bad rotator cuff problems, other things. And now lately I've just decided that balance is going to be there. I'm going to do that outlet. I'm going to take care of myself, because otherwise, you're just going to cut your ability to produce and to contribute to the field. You're really going to cut it short. The worst tragic thing is you don't do anything. You might cut it short and wind up with a bad heart attack or something, which runs in my family. So I have to take care of myself. So yeah, I think work-life balance is very important. But that stated, there's different definitions of it. I think anyone that wants to go into this field, and I see this with students often, graduate students, the ones that really struggle try to do it with a 40.0 hour work week. It's very difficult to make progress on a substantial question that you want to break ground on something, I'm not saying it can't be done, but you'd have to be incredibly well organized, very lucky, and have almost every experiment pay off at the bench. I see students do it and they've gotten their PhDs, but they don't wind up in the, they haven't published as many papers as other students, and therefore they're not as competitive. They're also not as well trained. So I think shoot for a 50, 60 as an average. I mean, it'll go up and down. That's what I recommend if you really want to do it. And if you don't, I mean, if you don't really have the passion for it, then I would say just, if you don't want that kind of a career, and it is a career and it is a major commitment, then just don't do it. Do something different. I mean, not like, I think, like I see my wife, she's an ARMP, she's a nurse practitioner. She works in the surgical oncology group at Shands, and she works pretty hard too. I mean, she's in there, has to be in there, sees patients, and she's often doing charts and updating stuff at night too. I mean, professions are work. They just are. I think there's very few people that can get by with 40 hours a week. So I think, yeah, the regrets is, and I think the last thing I want to say on that is, I think you work really hard as a postdoc, 68 hours a week, all the time. And now, whoa, I'm so jazzed. I got my own lab. Okay, I think what I didn't realize, and part of what they don't teach you in graduate school, is how to manage a team. You are maybe, I don't care how good a people person you are, it's very hard then to take on that role, that additional management responsibility. And I think my biggest regret is, and I've apologized to my first students. I had 4 students that started off my lab at A&M, and I was way too demanding, way too demanding. I figured everyone was going to be just like me. This is the way it's going to be. I think if I had to reset that, and I've learned that doesn't happen anymore, I am definitely much more tailored to individuals. Some people are better night owls, some are daytime people, some work, they can work more hours, some can't. My best lab manager I've ever had is currently Marie in my lab. She's got 2 boys. She has an assistant professor who just put in his tenure dossier. She comes in and she is organized. She works 40.0 hours a week. That's her job. That's what she's there for. And she's the best lab manager I've ever had. She comes in, so I don't need someone that works. So there's a perfect example of that. You broaden your horizons, and I'm not looking for someone that's willing or able to work 50, 60 hours a week.
Yeah, we have a question from Justin Schneider.
Next one from we got from Katie.
Oh, I think you sort of addressed Katie's about work-life balance. And then I was going to jump ahead a bit. Justin Schneider asked if you could give pointers on how you prefer a student to approach you on a research project. It's a question I think a lot of people will be interested in.
So where most of the students, it looks, I mean, I'm trying to figure out what's the enrollees in the course. You told me that some are on in attendance, some are going to see it taped asynchronously. But what's the range? Or like most of the students, what are we talking about, like freshmen or a mix, or?
It's a mix. It's a good number of freshmen, and then we have some people really all the way up to seniors who are kind of thinking about the next phase of their research career. So it's a pretty broad audience here. All
right. So let's see. So I think to address the people looking maybe for like freshmen or younger, or maybe anyone, doesn't matter what level, it could be sophomore, junior, looking for their first foray into a lab, particularly if you're trying to make a decision like I did. To be honest, my parents, they almost demanded that I go to medical school. I'm the first one from my family to ever have even gone to college, let alone all the way through a doctorate and become a professor and tenured at a university. Oh, tenured twice, I would add, by the way. Once at A&M, and I surrendered it to move to UF because they had a certain policy, and I got re-tenured again. So people look at this when I go give a seminar and they go, what? Tenured twice? that's a sign of how my sort of personal level of personal insanity. And people really tease me about that you would give up tenure and move to another place. I want to become a Gator so badly. And we did win the 06 championship 3 days after I got here. So why not in football? So yeah, so so I think, okay, the first thing is blast emails that are not tailored are not going to work. Okay. You've got to be willing to put in some elbow grease. Okay. So if you're really thinking about joining a lab, you should be excited enough to actually look at the papers that the lab has published, figure out what the lab has done, the general area, and become educated about it. And then you need to actually take time to write a focused letter, one that directly speaks to the professor and says that you've done that legwork. Because what does that do? I mean, there's a lot of students that want to go in labs. And I've learned over now my 25, 30-year career that of what to look for in people. And I'm looking for mainly enthusiasm. And enthusiasm in somebody that's willing to put in some effort. And that first thing that you see when those emails come in, I'll get in any semester, I get about, it seems like it's gone down lately, but I used to get 10 emails from 10 different students every semester. Okay, and I only have room maybe to take 1 or 2. All right, so it's a pretty tough process and it's very daunting. It's hard to get in the lab. I know this. My own daughter was trying to get into a lab at UF and I would tell her, well, I'm not gonna give you any nepotism stuff here. It's not gonna happen, but here's how you have to do it. Okay, and so she succeeded, but it took her a lot of effort to do it. And so yeah, so you gotta write something, I mean, something that tells the principal investigator, the faculty member, that you've read it and you're a little bit familiar with the area. They're not looking for like great insight and a new idea for a grant, like my student that's now been in the lab for a semester, but I think something to show that you did your legwork. Okay. And then what I always do is then I think having a resume attached right there, I think that'll help. All right. It's again, it shows a level of preparedness and that you can look at it. I think, you know, it is hard. I'm not going to say it's not for students that are trying to break through that have had no research experience. I remember this. I went my own self. I went when I was in a lot of your shoes as an undergrad. I went to a lab. He held up this thing. You ever use these things, pipette men? I says, no, I haven't, but I learn quickly. I pay attention, and you only have to teach me something once. Okay, I didn't get that job. I got a break because I beat out a couple medical students that wanted to do research, and I beat them out because I said that my parents wanted me to go to medical school, but I was really having doubts because I had taken molecular biology and biochemistry, and I was real excited about that and the prospect of doing research. And so I got my break first because they gave me an opportunity to say if it really stuck. And in hindsight, I'm glad he did because it did really stick, so to speak. I fell in love with it. I was completely enamored and I went for it. So I think that's the best thing I'd say about it. There is a little bit sometimes if you know another student that's in the lab, a peer, they might be able to identify somebody. They can ask people in the lab, do you know anyone that needs an undergraduate or something like that? That's another way to approach it, I think. But just try, keep trying, push.
Persistence pays off.
Yep.
Next question looks like is from Mohammed Yasser, asking if there's a skill students in biochemistry and related sciences need to practice before entering research.
I think science is an interesting thing. Biochemistry, molecular biology. I'm sort of a molecular biologist, biochemist. skill set. I got all the foundation I think I needed. I think the main thing is foundational knowledge. Anyone in any STEM field, I'm sure you realize you're here learning a foreign language. That's what you're doing, right? You're learning a way to communicate, technical terminology. I mean, I've got friends that are not in science at all, and they're fishing buddies of mine, and they work. One's a jeweler, the other one is actually constructs buildings, one of the chief contractors at UF that builds a lot of actually built the building that I work in, the Cancer Genetics Research Center. And so, yeah, they like listen. It's like, well, I listen to their foreign language. It's the same thing, right? Like, okay, yeah, like I know all that construction stuff and all what you're talking about, and they listen to me. It's just a sort of it's a terminology, a lexicon. You build it up. So I think what you need to do is build the foundation of knowledge that you need to in the area. And again, why that's going to become important is that first email I told you how to write it. You're going to have some of those terminologies in there. And then after the breakthrough, I'll interview 2 or 3 of the undergraduates, of which maybe I'll take 1 or 2. And then it becomes a little different. Okay. So I look for how, I don't know, let's see. For me, it's sort of, I think I'm not so, you don't have to be an expert in epigenetics, because you're not going to be, because you haven't been trained in the area. Okay. But you should know the basics. Like you should know like the vocabulary, like what a gene is, what a promoter is, what an enhancer is, the things that you can somewhat communicate in an interview with the faculty member. So then again, that's effort, but that's what you need. You need a basic knowledge. I would think at any university, and UF is definitely up there. I mean, I know what we offer in our Biochem 4024 course. You take that class, I think you can take that maybe when do you take it? Sophomore or junior? Take something like that, or a level course like that, or intro before it. And I think you'll have the basics of what you need to sort of break the ice with a faculty member. But I think also it's what you know. I mean, really, the more you know and the more hungry you are and the more information you accumulate and assimilate, the better off you'll be, because you'll be able to see different questions and where are there holes in the literature? Where do I go? Where's an exciting area to pursue further?
So there's a couple more questions in the chat, but while we still have a little time, are there any questions on the list I shared that you were particularly excited about answering?
Yeah, I mean, Elena's question, Elena Ashburn's question was, these were great questions, by the way. A couple of them I might not even want to bring up because they might make me sweat. Some of them are pretty intense, man.
You can skip those if you like.
I might. I like what actually caused me a lot of thinking. I'll probably, me, I'll just barrel in there.
Yeah.
So let's see here. So Elena asked, she said, you've been awarded many research grants throughout your professional career. Would you say that your educational experience gave you the tools you needed to know how to apply for those grants, or was it something that you taught yourself? All right, great question. All right, so grants, oh boy, yeah, the lifeblood of, and without grants, you don't do anything in this field, right? So, so I would say, so it's interesting. So as far as knowledge base, I think I've always been a tool person, and you even heard with Dr. Watson's introduction. I like to find a technology and I love it. I like to push the edge. Like, okay, so nobody could do that before. How do I do it? How do I devise a way to do it? So in graduate school, one of the things I'm really happy with the accomplishment is an enhancer is a gene regulatory segment that's way upstream of the promoter. It can be a kilobase to a megabase away. And somehow that element of DNA that binds transcription factors has to reach over and contact the promoter to stimulate transcription. Well, those were all ideas back in 1986 and 87 that people drew pictures, but nobody had a way to test it. And one day I just looked at it on the board And I said, oh, why don't we just do this? So you cut this ligate, and PCR had just started, and then we could do PCR across the junction. And that led to a whole series of technologies that we published the paper in Science, which is a very good paper because it was way ahead of its time. But it was, I think I got a real stroke of luck there that I was too far ahead of my time. And a world expert in this is Peter Fraser, who's at that other place, FSU, up the road there in Tallahassee. And he gave me one of my best compliments of my career. He says, Mike, that was just way too early for people, because people weren't really willing to accept it or recognize it for 9 years. But then it gave the birth of an entire field that was independently pioneered by another guy named Job Decker. Probably another regret of mine is that I didn't keep working on that. I probably should have kept going now that I think about it. But yeah, so let's see. So I think because I was so tool driven and trying to come up with new ways to do tools, I think I sort of hamstringed my own education during my PhD. I didn't learn effective question asking and hypothesis formulation. Like, because you have to ask questions in grants. There's a real bias at NIH grants, NSF. It's not as bad as it used to be, but you have to have a good hypothesis to answer to get funded. So I really think it was a self-inflicted wound. I could argue maybe that my mentor should have probably seen that I'm not getting that part of my education. So that I had to teach myself. And at the worst point, because I was still a tool guy through my whole postdoc, I got my job offers based on my tools. And then I got thrust into writing grants that were now all predicated on writing a good hypothesis. So I had to very quickly teach myself how to formulate a good hypothesis. All right. You could use the technology, but you still have to do it. So I had to teach myself that. But I think otherwise, I think my experience, you've got to go, if you're you're not going to be able to teach yourself how to succeed in getting funded NIH grants. You've got to go to labs where you get that training. And I picked a postdoc purposely because that was an environment. I knew how to write a grant. My mentor at my postdoc took the time, let me write grants. I had gotten grants on my own that I wrote. It was under him as a PI, but we didn't have like what we call co-principal investigatorship at the time. But I had direct experience in doing it. So I recommend you find that experience because you're going to need someone to teach that to you. And there are some good grant courses out there that teach you some good ways to say stuff. Simple stuff like, if you say you're going to do something, I expect to discover X. I expect to learn. I expect is like a good line between, oh, I'm going to do it, and coming off too like arrogant that it's a guarantee. Because if it's a guarantee, they're not going to give you the money. Because then it's too simple, it's too obvious, right? So there's a whole thing that we call grantsmanship. There's how to state it, how to phrase it, and how to just pose it to the, it's really it's a whole art. And I won't even pretend, I'm not nearly as successful as many of my colleagues. So, I'm still learning. So, but yeah, I think you need to learn that how to do that. Can't teach yourself how to write NIH grants on your own. If you can, maybe you can, but you're better, you're more accomplished than I am then.
Yeah. I think we have time for maybe one more question. So if there's one that I'll let you pick from the chat or the pre-written, what you'd like to.
Let's see. So what emerging technologies related to epigenetics am I excited about? And how do I see them impacting the field in the future? So it came up, and there's several questions are along these lines. Mm-hmm. And one was about, wow, hold on one sec. I'm having a battery issue for some reason. It got unplugged. All right. Whew. Thank goodness. Nothing like getting cut off there in the last question because your battery runs out. Yeah. Let's see, what was it? What am I so excited? There's a whole new field, and it's not just epigenetics and genetics, or they're two sides. Some PIs, some labs, they stick mainly to the genetic side, and some stick to epigenetics. And that's unfortunate because I think they go hand in hand. So for example, right now I'm working this mismatch repair gene. I found it's epigenetically silenced. We've known that for years. Well, by epigenetic silencing this gene, now you have error-prone problems in your genome, and you make a lot of mutations. All right, so they're very related, epigenetics and genetics. All right, so I think the most exciting thing is the cost of sequencing have come down so dramatically that we can see the first human genome, I think, cost $30 billion or $10 billion, I can't remember, to sequence the first human genome project. Okay, now we can do it for about $800. We can sequence an entire person's genetic complement. And now we're getting ways that are coming down epigenetically. The first machines are coming out that we can also get someone's entire epigenetic profile, the DNA sequence, and all the methyl groups that are later on the DNA, plus the stuff I can add as well at the same time. And that's coming down to the $1,000 range. So I think the most exciting thing is the days of like treating by probabilities, let's say patients with cancers, and saying, well, this works off in this. Now they're already doing it. They can pattern, they can look for known DNA mutations, and they can tailor those treatments to each patient directly, individually. There are certain things that you can get that information, genetic and epigenetic, and I could take that, and then I can say, well, I've got no business at all as an oncologist giving this patient this chemotherapeutic regimen, because all I'm going to do is give them bad adverse side effects. I'm going to damage their DNA further, and I'm not going to be able to cure them. So a personalized medicine approach is definitely coming. And I think the most exciting thing about what's coming out is immunotherapy is huge. I think anyone thinking about an area, retasking, cellular engineering, training your own T cells in your immune system to attack your own tumors, that's the wave of the future. Because hopefully in about 20 years, we look back and say that oncology was a sort of realm of barbarism, because like I said, these chemotherapeutics, they're usually DNA damaging agents. They're very toxic. They cause all these things like making you nauseous because it kills your intestinal lining, your hair follicles get killed and your hair falls out, all these things. Immunotherapy is great. You can, your own immune system, your T cells get activated, your natural killer cells, and you kill the tumor cells. All of us are doing that all the time, every day. And I think strongly what's emerging is that cancer breaks through when you're immunocompromised or something. That could be anything from stress to whatever. So I think retasking the immune system, that's a really great area. That's what I think is going to be exciting. All right.
Exciting. Well, thanks so much for joining us and giving us your time. This has been great.
My pleasure.
Really appreciate you taking the time. We know your schedule
Thanks
is
again
quite
for
busy.
having me. If anybody wants to email me, it's real easy to find me. Kladde, Google, I'll come up first, and Kladde at ufl.edu. That's it. Simple email.
Fantastic.
All right.
And they
Okay.
have shared your faculty profile and some other links with them in preparation for this, so they should be able to find your information easily. But thanks so much.
Great. Thanks again for the invitation and the opportunity to put myself out in front of you all.
Awesome.
Thanks
We
again
really
for the great
appreciate it.
questions. Bye-bye.
Bye.